different chat places. One on the link that I sent out, and then this live stream went to a different place. Sorry, the pains of a first timer. So I hope that the guys from the other room got my message and moved to this one. There are 20 something of you guys here, so I said this when there were fewer people here. Say hi in the comment box so others know you're here, and also any questions that you have, take this time to flip through your lab book if you have it in front of you, or if you can think of some obscure thing that you are not understanding, just type it in and I'll try to figure out and get us both on the same wavelength. I'll start with Kira. You asked, what about the dilutions? Will it always be multiplying by 1 over 100 at the end? The answer to that is the 1 over 100 represents the calibrated loop. So that little yellow loop that came in the sealed packaging that we used in lab. So if the question that is asked specifies that you use a calibrated loop in order to plate, then yes, you will use the 1 over 100. If it doesn't specify this, you will not use the 1 over 100. Now we have a question from Josie. You're confused about the calibration factor on page 16. You have objective 4x, and that equals 25. I think I remember what you're talking about. On page 16, just above exercise 1, there are the calibration factors next to the ocular micrometer. The way that we use this is that you have your microscope, and if you look on page 15, there's an image of the microscope, and then you have your oculars, which are the little eyepieces that you look through. By the way, you need to know the parts of the microscope for the final, because there will be a diagram of the microscope, and you will have to label different parts of the microscope depending on whether Catherine asks you to label like D and E, or she asks you to label the empty spaces. So back to Josie's question. The way that we gauge the size of what we're looking at through the microscope is what the ocular micrometer is used for, and I think this is where you're going with the calibration factor question. So your 4x objective equals 25. That means that when we're looking through the 4x objective of the microscope and you see something that falls between 2 notches of the ocular micrometer, the little ruler inside the ocular, in order to get the true size of what you're looking at, you would multiply 2 by 25 to get 50, and that is the actual size of the bacteria or object that you're looking at. In the same way, if you look at the same thing, the same, let's say, microbe through the 10x objective, so you've moved from 4 to 10, in order to get the true size of what you're looking at, you would take the number of notches that bacteria is on the ocular micrometer, the little ruler, and you would multiply that by 10 in order to get the true size. So let's say it fell between 3 different notches, you would multiply that by 10 to get 30, and then the true size of that is 30 micrometers. So Tessa asked if I could go over the steps for aseptic technique. So I'm going to do that now. So aseptic technique, as it applies to the lab exam, begins with your lab entry exit protocol. which is in the Lab 1 slideshow. It starts with when you enter the lab, you put your supplies on the seat bench. So I would recommend that when you come into the station for aseptic technique at the exam, you begin by placing your papers and any pencils or pens that you bring with you into the lab exam. You place them on the seat at the station. If there isn't a seat at the station, I'll try to make sure that there is. But if there isn't, find somewhere else to put them behind you or something, because that's step 1. Step 2 is to properly wash and disinfect your hands. Make sure, because the station is at the sink where there is foaming soap just for washing your hands usually, make sure you use the proper soap to wash your hands, the antimicrobial soap. Then once you've washed your hands, you put on your lab coat. There will be lab coats at the station. If you feel more comfortable just wearing your lab coat to the lab exam, you'll feel a little bit silly because you'll be moving from station to station, writing on a piece of paper and wearing your lab coat, but it is a way of getting around forgetting to put your lab coat on, which is something that I've seen some students do at these lab exams. Then you're going to disinfect your bench with 70% ethanol. Make sure that you have continuous contact with the bench. Remember, this can be a question as well, the continuous contact of the disinfectant with the bench. You can do this. There will probably be rolls of paper towel at each station, but you can also do it with some paper towel from the hand washing station. And then you'll begin your aseptic technique. So you're all ready. You have your loop or your needle if you're doing a deep, and you're going to, let's say this is my needle. So you're going to have your needle, and what can I use? This wonderful nail polish will be my inoculating tube. So you take your needle and you flame your needle. Remember to wait until it turns red. You pull it out, don't wave it around, wait for it to cool, so count to 5 or something. Then you're going to use your pinky finger and your ring finger to pick up the tube off of the bacteria tube. You're going to take your tube of bacteria and flame it against the incinerator. Pull it out. Now you have your loop. You're going to dip it into the bacteria, pull it out, and then don't forget to put the top back onto your bacteria. Put it down. Yes, Josie, you're right. Flame it again before closing it. Always flame the tube. I keep forgetting that. You guys are so much better at this than I am. At this point, you have bacteria on your loop. You pick up your inoculating tube or your tube that you want to inoculate into. You place your loop into it, shake it around a little, pull it out, put the top back on your tube, flame your loop because there's bacteria on it, pull it out, and place it back in your test tube rack. It's more than likely that you will be inoculating plates. So you're going to choose the correct plate to inoculate and follow the aseptic technique of disinfecting your bench, flaming your loop, waiting for your loop to cool down, opening your tube of bacteria, flaming it, collecting your bacteria, closing the tube of bacteria, placing it back in the test tube rack, and now that you have bacteria on your loop, you're going to take your plate with your media on it, open it, inoculate in a T streak, close it up, flame your loop, put your loop in the test tube rack, and then you're going to have your inoculated plate. Remember to label your plate properly. Sometimes the instructions that Catherine puts up remind you to label your plate, but if after you disinfect your bench, you label everything, it'll be a lot clearer for you. Once you're done, don't forget to disinfect your lab bench, then remove your coat. You're going to place the coat back on either the chair or the hanger, wherever Catherine originally provided them for you, and then proceed to properly wash and disinfect your hands, pick your things up, and then you'll either be placed at a waiting station or the timer will ring for you to move on to the next station. I hope that was thorough enough. Oh, guys! The professor is with us. Hi, Bob. Josie, your question was, should we know all the different characteristics for slant, broth, and deep? And no, I'm not in my pajamas because I had to do my master's presentation at seminar today, so I actually had to look presentable. So yes, you do need to know the different characteristics of slant and deep, but the characteristics are not so much, they're not so complicated. So you have your liquid soy agar, your liquid soy broth. The agar refers in this lab more to the solid form. So the tryptic soy agar plate, this is a solid. You should know that a slant and a broth are inoculated with a loop in opposed to a deep that's inoculated with a needle. When it comes to the results of the slants, you do have the beaded, tree-like, straight, diffuse spreading, rhizoid type of growth. So there might be, it's a little bit of a cruel question, but there might be a question asking you like to name 2 or 3 of the forms, or maybe there'll be a photo, like a diagram of it asking you to label which one's which. Josie, as to your question, you should know that the filiform is the straight form and the arborescent looks just like the filiform, except there are branches coming out of it, so it looks more like a tree. So yes, the diagrams, if she puts them on the exam, the diagrams will be very clear. Then you have your results for your colonies on the T-streak. So if you're looking at a TSA plate that has a growth on it, is it a circular growth? You might be given a plate with a very clear large colony growing on it and asked what are the margins. And I just noticed my dog is screaming in the other room. There must be a fox outside. 1 second. Cassam, come here. Come. Come here. Come. Sit down. Quiet. So Grant, in the first PowerPoint for Lab 1, there's a page of results. This is also available in your lab book, where you have the shape, margin, elevation. Hey, come here. Come. Right there. Lie down. Here. Sit up. Come here. Lie down. Don't be stupid. Sorry, she's being very, very loud. Then you have your size of colonies. I doubt that she'll ask about this because it's pretty easy. There's like punctiform, small, large, moderate. Now we get into the deep inoculation interpretation. So Remember, you do have your obligate aerobes, your anaerobes, your aerobes, and your facultative anaerobes. And you need to know how the growth throughout affects that. Of course, like I said, we've been over oxygen requirements so many times that I feel like it would be impossible for this to not be a question. So definitely go over oxygen requirements. So if it's growing throughout the test tube after you've inoculated with a DEEP, then it's a facultative anaerobe. If it's only growing at the bottom of a DEEP inoculation, then it's an obligate anaerobe, because if it's exposed to oxygen, it dies off or it's not capable of growing. And then you have your obligate aerobe, which if you inoculate a DEEP, will only grow at the top because it needs access to oxygen. Things like this are definitely fair game. Teresa asked if I would be able to list the important types of agar plates that you guys should be able to differentiate. I brought some of them. Some of them have run out in the time that we've used them. I stole these, and by stole I mean got permission from Catherine to take them from our containers of lab. Only some of the media were still left and ready for me to take. Here is a starch hydrolysis plate, an SHA, and here's a TSA, which is tryptic soy agar. So as you can tell, they look almost identical. It's very hard to tell them apart, but the thing is that sometimes Catherine will leave these labels on them, and then you'll be able to tell the difference. You've got your MSA plate here, and I'm going to put it by the EMB plate so that you can tell that they're a little bit different in shade. Again, they're labeled up here at the top. These labels might stay there, or they might be erased. But it's not so much important that you be able to recognize them and simply know what they are based on what you're looking at. It's more important to know what kind of tests you would run on each one of these different plates. And the final plate I have here is the blood agar plate. This one is really easy to identify because it's a very solid red, unlike the others that are a translucent red. And again, you should know what kind of a test you would run on a blood agar plate. She won't place these in front of you on the exam and say, identify these plates. But for the streaking station, there will be a little note, and it'll say things like, inoculate a blood agar plate with blah, blah, blah. And you'll need to know out of the 6 or 7 different plates that you have in front of you, which one is the agar that you're looking for, and inoculate that one. So you will need to know how to identify them based on that. Adele asked about the question in the lab manual, I'm just going to read it, whether or not the lab disinfectant and the chemicals had a fair chance of killing the bacteria in the modified yeast dilution. So if you're talking about how we, looking at the results from lab 4, that the only thing that was effective at killing the bacteria was the lab bench disinfectant, and not the janitor's cleaner or the diluted hand soap. Grant asked if the soy agar plate and the starch hydrolysis plate both at the streaking station, if they're both at the streaking station, will they have the stickers, or will you have to be able to tell? Most likely, if they're both at the station, the TSA plate is labeled, or they're both labeled. I highly doubt that Catherine will trip you up by removing the labels from all of them, especially when they look so identical. But while we're on this topic, it's important to note that if Catherine is telling you to perform a starch hydrolysis test, she won't specify to use a starch hydrolysis plate. So make sure you use the right plate for the right streak, given the instructions. Shaylin asked if I could show you the difference between the MSA and the McConkey agar plate. The answer is no. This is the MSA plate, but I couldn't find any McConkey agar plates left in the boxes we keep the extra plates in. But I can tell you that the color difference really, it's not very easy to tell. Focus, don't let this freak you out. Focus on what is an MSA plate used for and what is a McConkey agar plate used for. Like, what kind of tests are we running on them? And based on that, you'll be able to tell if you should use an MSA plate or a McConkey agar. If the streaking station has both, I'm sure one of them will be labeled. In lab number 2, Catherine showed us a plate of a contamination. This is a legit possible question where she could put a plate down that has been T-streaked with an arrow pointing at a contaminant and ask you what that is. The question could be a multiple choice saying things like, this plate was streaked with K. rhizophila and Ecoli. And then she's pointing an arrow at the contaminant and asking, what's this? And the possible answers could be, this is E. coli, this is K. rhizophila, this is a contaminant, or it's unknown what this is based on the information provided. And of course, if you can recognize a contaminant, which is something that doesn't look like anything else you've streaked on the plate, then you're golden on that one. Looking at the microscope, we went over the definitions for par-centered and par-focal. These are fair game questions for the exam. So when you change from one objective lens to another and the image stays centered, so when you change from 4x to 10x and the image that you're looking at stays centered but not necessarily focused, then it is a par-centered situation. But if you change from one objective to another and it remains in focus, that's par-focal. And the difference between the two could either be a question with a blank area or it could be a multiple choice question. The process of putting your microscope into an oil immersion is almost 100% guaranteed to be a question. Either you'll be asked to arrange the steps, so you'll be given a jumbled up list and asked to put 1 through 7, because there's 7 steps, 1 through 7 in the correct order. So make sure that you know that. The oil immersion lens steps are, and if you did this in lab, it's in your head, you got this. So first you're going to be at the 40x and you're going to swing it halfway, so you're in between the 100 and the 40. Step 2 is place a drop of oil down. Step 3 is to swing the 100x objective into the oil. And then step 4 is to use only the fine focus, because if you use the coarse focus, you're going to be moving the stage too much, and you might bump the lens into the stage, and you don't want that. So use your fine focus. Step 5, I don't know why step 5 is here, but it specifies about a cover slip and how it's not necessary during the oil immersion stage. And step 6 is if you drag the lens through the oil, you need to clean it away. And step 7 is to clean the objective, the 100x objective, when you're done. You could simply, if it's just a question for you to answer, make sure you mention that you swing out the 100 halfway between 100 and 40, then you use a drop of oil, then you swing the 100x into the oil, use your fine focus, and then state that at the end you'll use lens paper to clean away the oil from the objective. Mark asks, Mark is inviting everybody for beer on his dime, and he asks, just kidding, if you need to know the bacterial colony characteristics. The list from Lab 1 about your bacteria colony characteristics is fair game. About the margins and if it's elevated and how large it is, all of those descriptive words are fair game. I hope that answers your question. You could be asked about the process of heat fixing. So remember, we prepared smears in Lab 2. We placed broth onto the slide. We added water when the bacteria was collected from a slant because it was a lot thicker versus collected from a liquid broth. And then the bacteria was allowed to dry. Once it dries, you heat fix it. You don't use the heat to dry out the liquid. Okay, stains. I was asked to go over stains. The question I remember from last year's exam is about a background stain. So Catherine provided printouts of different bacterial colonies that were stained, and the nigrosin that stains the background and leaves the bacteria clear. So that photograph could be placed there, and you could be asked, what kind of stain is this? And if it's a multiple choice question, she'll put things like gram positive, gram negative, background stain, or let's say a spore stain. And so if you know that nigrosin is a background stain, then you're good to go. Know the difference between differential staining and background staining. So in differential staining, and gram stains are a differential stain. A differential stain stains the bacteria directly, while a background stain stains the background. So an example, if she asks for an example of a differential stain and an example of a background stain, your nigrosin is a background stain example, and your gram stain is a differential stain example. Next. So then you've got your gram stain steps. This is a fair game question. It's a difficult question, but it's fair game. The steps of getting a gram stain done. So you're placing of crystal violet, and then iodine, and then ethanol, and then safranin is, unfortunately, there are some things in science that we just need to memorize, even though if you were working in a lab in the future, you'll probably have the protocol in front of you, so you won't have to memorize it IRL, but for this exam, it's a legit. In your lab books with a picture of a spore stain and a picture of a capsule stain. These are stains that we can't do in labs. Be given a photograph of the spore stain and the capsule stain, and be asked which one's which. Or a true or false question, which is a capsule stain will stain both the bacteria and the capsule the same color, true or false. Or maybe it'll be a picture of a spore stain with an arrow towards the blue spores and an arrow towards the orange bacteria. This is the picture you have in your lab book. That's why I'm using those colors. And you'll be asked, like, what is A, what is B? Another example of a differential stain in addition to the gram stain is the acid-fast stain. This is one that you did in lab. Mannitol salt agar was used in lab 3. This is an example of a selective media, so that could be a question. Give an example of a selective media. So the mannitol salt agar will change color back to the regular agar color, so back to the TSA color, once a certain bacteria is grown on it. So the reason it's a differential media is because it can differentiate between bacteria that are able to ferment mannitol, mannitol salt agar, and those who cannot ferment mannitol. So if a bacteria is able to ferment mannitol, this mannitol salt agar will go back to being the color of a soy agar, and if it's not able to ferment, it will remain pink and the colony will grow on it. However, when a colony doesn't grow at all on the mannitol salt agar, such as Ecoli, it's because it's being inhibited by the salt concentrations, so Ecoli can't grow at all on here. You could streak this whole plate and all you'll see is your streaking and not the colony growing. So it's able to differentiate between bacteria that can ferment mannitol, cannot ferment mannitol, and colonies that are inhibited by salt. McConkey's agar, which I couldn't find, that is the agar that contains lactose, salt, and a pH indicator. So this is also a selective media. So it selects for gram-negative bacteria, but it's also differential. So this is an example of a selective and a differential media. This is the McConkey's agar. It differentiates between the bacteria that can ferment lactose and those that cannot ferment lactose. Unlike the mannitol salt agar, the difference between those that can ferment mannitol and those that cannot ferment mannitol, and it selects for gram-negative bacteria. If a bacteria is able to ferment lactose, the pH will drop, and as the pH drops, the red is absorbed by the bacteria. So the line that you streak will appear bright pink. So the bacteria that you streaked will be bright pink and be surrounded by a kind of darker color in the agar. If a bacteria is very much able to ferment the lactose, it will not only produce like a pinkish color, but the salt that it absorbs will be put out to form a halo around the bacterial colony. If a colony is alpha hemolytic, it'll produce green halos. You'll see green halos around the colonies. Let me open this. Green halos around the colonies inside the plate once you've T-streaked it. Of course, your zone 1, you'll see nothing. Zone 2 will be harder to see, but once you get to zone 3 and your T-streak, you'll see little green halos around the colonies, and that's the alpha hemolytic. They will consume the blood from the blood agar plate. A beta hemolytic, these are able to grow a little larger, and they will also consume the agar on the blood agar plate. A gamma hemolytic or a non-hemolytic. I don't like gamma hemolytic as a name because it implies that it is hemolytic. These non-hemolytic bacteria are not able to lyse the blood, and so you'll just see the bacteria growing on the agar without having changed the color at all. So remember we talked about oxygen requirements in Lab 1. We talked about anaerobic, aerobic, and facultative anaerobic. Well, there's 2 more. I think there's 3 more, but there's 2 more that we touched on specifically in lab. The microaerophilic and the aerotolerant. So microaerophilic looks a lot like aerobic, except it's not all centered at the top of the tube. It's got kind of like a band, and then it goes down and becomes a band of bacteria just slightly removed from the top. Microaerophilic, somewhat liking of air, if you break that word down. The aerotolerant would look a lot like the facultative anaerobic, except instead of it being centered at the top and then diffused throughout the entire deep, it'll be simply diffused throughout the entire deep equally without the accumulation at the top, and that's the aerotolerant. And then you've got your 4 tests that you did in lab 3, which was your citrate test, your starch hydrolysis, your MRVP, which is your methyl red Voges Pasteur test, your direct test for fermentation of a single saccharide, which is where you used, I believe it was lactose, sucrose, and glucose. A citrate test, you should know that a positive citrate test turns blue, a negative citrate test is green. Okay, and then you had your starch hydrolysis plate, which is this one, where you streaked it, and then you incubated it, and the test itself came from adding iodine solution to the plate afterwards. So knowing that process, knowing what that process tells you, if you add iodine to a starch hydrolysis agar plate, and you see that the bacteria streak is there, and then the iodine covers the entire background of the streak, then it was not able to hydrolyze the starch around it. But if you streaked the bacteria and then put iodine on it, and there's like a halo around the streak that you made, that is an indication of the bacteria's ability to hydrolyze starch. We'll start with the MRVP, which is, as you remember, you prepared 4 tubes exactly the same. The difference was that you inoculated with 2 different bacteria. So you need to do your your studying on what the different tests are used for, the MR and the VP. Okay, so the fermentation of a single saccharide, we found positives. If they changed color from the red, they began as red, right? And if they changed color to yellow, it means that the saccharides were fermented, so they were used up by the bacteria. In lab 4, you used gelatin, and understanding why certain bacteria were able to hydrolyze the gelatin and why others weren't is important. So if a gelatin became liquid, is that a positive or is that a negative test? Hint, if it's gelatinous, which is like a gummy bear is gelatinous, and it becomes liquid, that would be an indication of a positive test. If it remained gelatinous, that's a negative test. Sorry. Then you did your test for motility, whether or not a bacteria has flagella and is able to move around. So for this example, I have my trusty little fuzzies here. So as you can see, coli has flagella and it's able to swim around, so it's motile. And tuberculosis is just a rod bacteria, so it probably won't have motility unless the producers didn't put flagella on the tuberculosis on purpose. Go back to the MIO test, the motility. Now remember, you can't just put random letters on the test and get partial marks for it. So you need to understand that the MIO is a motility test. You need to understand that your MRVP are two different, like MR and VP are 2 different tests. You can't just put MRVP MIO because I have seen that before. So if we go back to the MIO test, your indole is the red and yellow layers. So is it positive or negative for indole? So after you add the Kovacs reagent, it tells you whether or not it's positive for indole, my bad. And then your motility is given by the change in the haziness of the media versus the lack of haziness. And then remember, it was very hard to see in labs, so I recommend you download and look at the PowerPoint so you can see the photos. But the photos show ornithine decarboxylation in gray at the bottom of the tube. So the MIO does have 3 things that it tests for, and that's fair game on the exam for Catherine to ask you a multiple choice question, asking like, what does MIO test for? And the first couple answers will just have like 2 of the 3 in each one of them, and the third answer will be, let's say, all 3 of those tests. So remember that the MIO tests for ornithine decarboxylation, and that's shown by the gray haziness at the bottom. It tests for indole, which is reached by the Kovacs reagent at the top of the tube, and then the motility, which is the haziness within the tube. On to the nitrate test. Now this was one of the things on the list that people asked me to touch on. So if a color change occurred, that means that we didn't have to add zinc. So the color change occurred, meaning the nitrification happened all by itself. So that bacteria was able to nitrify within the broth. And then when we added the zinc, if it changed, then it means that the nitrate wasn't nitrified. And then if it didn't change color at all, that means that there was nothing for the zinc to react with, and it was already passed into the ammonium forms of nitrate. So there was nothing there to react with the zinc. I don't know if Catherine's going to ask about the nitrate reagents. I think it's a little benign to know that the 2 reagents that we used in the nitrate test were sulfonilic acid and dimethylnephthalamine, but if you're able to commit those to memory, I recommend you do. An oxidase test. Who can guess what this test was used for? We use this to determine cytochrome C oxidase. So it's important to know that cytochrome C oxidase transports electrons from donor compounds such as NADH, NADH+, to electron acceptors such as oxygen. Catalase test was positive by bubbling, so a possible question could be, how do we determine a positive catalase test? And the question could be the answers could be change in color, change in liquid form, no change in color, or bubbling, and obviously the answer is bubbling. The demonstrations for moist versus ultraviolet heat, those are fair game for the exam, so questions like what was more effective at destroying bacteria, moist heat, ultraviolet heat. Of course, there will probably be that question about what form of disinfectant or destruction of bacteria is most effective, and we did look at that chart that showed that moist heat, unlike dry heat, so moist heat is autoclaved, so the autoclave does create a very hot, moist environment that kills off almost all the bacteria that we use in the lab. So that would be the most efficient way of killing bacteria, because it both dissociates different proteins as well as kind of destroys the cell walls of the bacteria. Remember the antimicrobial drugs used. What I would focus on here would be the zones of inhibition, what they mean about the bacteria. So if Catherine asks something or shows you a plate that has, let's say it has bacteria, like Ecoli streaked throughout the plate, and it has 4 different antimicrobial discs on it, and you're seeing zones of inhibition in 2, she could ask you, what can you tell me about the ability of the discs A, B, C, and D's ability to kill off E. coli? Or she could ask, which one of the following discs A, B, C, and D are effective at inhibiting the growth of E. coli? Adele, I don't think you need to know the exact number of each zone of inhibition, but if there's a very small zone of inhibition versus a medium or a very large zone of inhibition, understanding why that's happening would be a plus. it's not something that I would be super focused on, memorizing numbers to sizes of zones of inhibition. I hope that answers your question. Ornithine decarboxylation, which was tested in those pink tubes that we added the Kovacs reagent to, a negative reaction, and what it means is that the glucose in the medium, in the pink media, is fermented, which causes an acidification and creation of the yellow color at the top when you add the Kovacs. So knowing what a negative reaction looks like and why it happens is important, because if there's a question in the exam, it'll be about that. And a positive reaction is the ornithine decarboxylation happened anaerobically, and it produces an alkaline, unlike the negative, which is an acidic reaction. So it produces an alkaline reaction in the positive, which overcomes the acidic fermentation, and that's what causes the purplish blue color in the media. So as you can remember, I'm pretty sure that the 2 bacteria that we used caused positive reactions. In lab 5, in lab 4 we touched on the mesophile bacteria and what that means. Do know the different terms for these. So mesophiles are bacteria that can grow in 20 or 30 or 37 degrees Celsius, meso in the middle, and then you've got your extremophiles, which would be either your extreme cold or your extreme hot, but your thermophilic, heat loving, would be the ones that would grow in the extreme conditions, and these definitions are fair game. I don't know if you've probably touched on them in class as well, so that'll help solidify that in your noggin. Morgan asked, what temperatures do thermophiles grow at? And without looking it up in a definition and going entirely from memory, thermophiles are extreme heat ones, so I would guess something that's supposed to kill off a bacteria is at 60 degrees. It's supposed to cook it completely, so I would say anything approaching or after 60 degrees. But I would recommend to look it up in your textbook to see what answer they're going to go by on the exam. Okay, dilutions. If you have a 10 to the negative 7 dilution, you would put 1 over 10, because you took 1 mil of your original substance into the first 9 mils, and then you took 1 mil of that mix into the subsequent dilutions. So if you have a 10 to the negative 7 dilutions, you have 7 of these. And so you would put 1 over 10 to the power of 7, because you did 7 dilutions. And that number would give you what you would need to multiply your colony count by in order to get the original number of colonies in the original milliliter that you removed from the original substance. Okay, Josie asked that I do an example including the calibrated loop, so I'm going to do that one. Let's pick 3. So 10 to the negative 3, you have 1 over 10 to the power of 3, and then you multiply that by 1 over 100. Your calibrated loop is 0.01 mils, so 0.01 mils would be 1 over 100. If your calibrated loop was 0.001 mils, then you would have 1 over 1000. I hope that straightens that out. So 1 over 10 power 3 times 1 over 100 gives you 1 over 100,000. And then let's say you counted 5 colonies on your plate, you would multiply 5 times 100,000 to get the true number of colonies in your original substance sample. Okay, Josie, so I think that what you're struggling with is how that 1 over 100,000 becomes the 100,000 number. The 1 over 10 power x times your calibrated loop, that number, the reason we do that is to show you how we arrive at the number that we multiply the colony count by. So it's illustrating for you how we arrive there. Then you can just ignore the 1 over 100,000, just look at the 100,000. One second. I have to go let the dog in because she's barking up a storm outside.
Okay, I will get.
You got her?
Yeah.
Oh. You got her. Pipettes. So in Lab 5 we did use pipettes. So the two diagrams that are labeled that I want you to focus on knowing the components of all of them are the microscope, which is, it's legit for Catherine to put down a picture of a microscope, point towards something and ask you to name it. In the same manner, I want you to do that with the pipettes. So there is a diagram of the pipettes in the presentation, so take a look at that and look at what is the push button, which also happens to be the large adjustment knob, where's the tip injector, where's the wheel for fine adjustment. So you guys didn't use the large adjustment and the fine adjustment because they were already calibrated to 90 and 10 micromils for you. But the fine and the large adjustment knobs change the volumetric number that's displayed. So understand that that display displays your volumetric number. Your tip injector button connects to an actual tip injector, so know what that button controls on the pipette. Then you have your shaft, which is pretty self-explanatory, pun completely unintended, and then your disposable tip and where it marks onto. Know the unbreakable rules of the pipettes, just in case Catherine throws them on there for bonus marks or something. Understand how to draw parallel between the micro dilutions that you did in Lab 5 and the regular 1 to 9 mil dilutions. So you used a 10 micromil and 90 micromil pipettes. How does that translate into your regular dilutions? And try to draw a parallel between them so that you have an understanding and don't have to learn two things separately. What else is important? Okay. I can't really explain the most probable numbers because it won't be very effective in this format, but I do recommend that you go into the section where we talked about brilliant green, 2-time brilliant green and 1-time brilliant green, or as I was referring to them, brilliant green and less brilliant green, and then look at the table on the next page so that you can understand your most probable numbers based on the numbers of positives that you got in each one of those tubes. So that brings us to the end of the 5 labs. Let's just look at the things that were written on the board for me to review and see if I've touched on them all. So we did aseptic technique already. We went through stains, so differential stains, background stains. What are they? You should definitely go through that and figure out examples that you can name specifically for each one of them. Spore stains, capsule stains, differential stains, background stains, what kind of a stain is a gram stain, what are the steps for staining a gram stain. Other things that were written on the board were different media and why we use them. So we went over the different plates that I've captured here, and we talked about the different tests like starch hydrolysis and what else? MRVPs, and what media, what those tests can tell us through the media that we developed them in. Incubated them in. What else is on the list? Membrane filtration technique, someone wrote that down. I don't know if that is still something that people want me to talk about, but pretty much what we did, the purpose of why we did it is that you chose between sewage water and fountain water, and then you placed 50 mils of that sewage or fountain water and you filtered it through. Then you used sterile water, which I hope in an ideal world it should be autoclaved water. So the autoclaved water was used to wash through the water that you originally chose to make sure that all the bacteria that was contained within it made it onto your filter paper, and then you incubated that bacteria on a tiny, adorable EMB plate. So that was the purpose of the membrane filtration technique. I haven't seen any of the plates yet that you incubated, so I don't really remember what the results were from last year, but pretty much you were growing bacteria. You were filtering through a certain water type in order to see what bacteria were contained within it, was the purpose of the membrane filtration technique. Okay, so I'm going to sign off now and go eat dinner with the man who pretends that I don't exist right now because YouTube is embarrassing. Yeah, alright, so I'll see you guys at the exam or next Friday for another one of these. Bye!